in addition to producing fresh arm type 2 and arm type 1 cells, is the major treatment and impact of mesenchymal stem cell treatment in the lung. And here's an example of our data and other people's data as well. We have three groups of animals, group A, B, C. In A, this is a normal animal. And you can see the lung is beautiful. We have a lot of air sacs, which perform or alveoli, which perform the gas exchange. And when we stain A with some methodology for collagen, like mesenteric role, you can see there is not enough collagen. There is no increase in collagen. However, when you expose this animal to the polymyxin, when you treat this animal with polymyxin, they will grow the symptom similar to the fibrosis. Therefore, this animal produces more collagen. You can see collagen in E. B is eosin hematoxylin, histologen section, and the E is a collagen. So you can see increase in the collagen. I refer to it using the yellow arrow. In the third group of animal, we expose them to polymyxin and then inject the mesenchymal stem cell. And you can see the big difference. The amount of collagen is reduced to the minimum. You still have more collagen, but the amount is reduced. In addition to that, there is a much more improvement in the number of alveolar type 1 cells and alveolar type 2 cells as well. So it is a really effective method in animals. The third example of the application of stem cell therapy in the treatment of respiratory system diseases is the use of induced pluripotent stem cells. You know that induced pluripotent stem cells, we got it from any part of our body, like from the skin, and induce their pluripotency by activation of some gene like NANOC or OCT4. So we got IDSC. And we can inject this IBC into our animal model of some diseases like bronchopulmonary dysplasia, and we can see the difference. You can see the much improvement in this animal model. And here is an example. If we have three groups of animals as well, normal one is A. You can see beautiful structure of the lung, a lot of septa contain alveolar type 1 cells, and a lot of air sacs. This is beautiful structure. When we expose the second group of animal into hypoxia, increased oxygen. So now there is a big damage in the lung, similar to the bronchopulmonary dysplasia in humans. This is our model. In the third group, we exposed it to hypoxia and then treatment with MSC. And you can see major change in the structure of the lung simply because we have more production of alveolar type 2 cells, more production of alveolar type 1 cell, more functional lung. So it's a really effective method. So this is the three examples of how we use the stem cell for the treatment of some major or important respiratory system diseases. So let's go now about the general application of stem cell. The most commonly used stem cells for the treatment of human disease and also for clinical trials are mesenchymal stem cell. As you can see this in chart, you can use mesenchymal stem cell for clinical trials in bone and cartilage diseases, in heart diseases, in respiratory system disease, and in GI or gastrointestinal disease. They can be used in this application. And actually, this fast growing research, we have annual growth rate of 77% since 2008. We have this annual increase since 2008, 77%. We have hundreds of applications, if you go to PubMed, on this topic application of stem cells and their clinical trial as well. And here is a chart example of how can we use this mesenchymal stem cell for the treatment of one system, like respiratory system. We are using this mesenchymal stem cell for clinical trials in PPD and another disease called RDS, in IPF, and many diseases. So it is really highly productive area and fast growing area of research. So let's now move now to the last part of my presentation about stem cell as an industry, stem cell as a market. So stem cell industry is a rapidly growing biosector. It is a part of biotechnology and it is rapidly growing. The global stem cell market worth 15 or over 16 billion US dollars. So we have actually, the number is more than that, but this is a figure what I have. The global stem cell market size worth $16 billion. And stem cell segment is a fast-growing biotech industry and can alter the treatment of the world diseases and also discovery of different medicine and drugs. So let's move to the Egypt. How about Egypt? So Egypt, similar to other developing countries, there is a limited stem cell use and there is a limited stem cell market, simply because there is a lack of practical regulation like other market and developing countries. I live in the USA and UK for over 20 years. And also I visited some developing countries and I can now compare. So the problem in Egypt, similar to other countries, is that we have a limited stem cell use and stem cell market simply because there is a lack of practical regulation and lack of investment. So Egypt stem cell therapy are developing slowly, and there is a lot of or many research difficult. So based on my experience, I can suggest this agenda to promote stem cell industry for diseases in Egypt. Stem cell industry in Egypt can be promoted by more supportive regulatory policy to drive further growth, and also by more private investment. We need a private sector to invest in this area to help the stem cell industry to achieve its full market. And let us take the success story of other countries. Other countries, the stem cell industry are growing fast in other countries simply because they are more focused on commercialization of private and public research, and we have more formulation of regulatory framework and public interest. So in my last slide, I'm going just to suggest some idea for Egypt to follow. So the Egypt stem cell therapy can be improved by attracting more organizations to invest. We need more investment, especially from the private sector, and also to create and commercialize new technology. We can't cover everything. So my suggestion to my mother country, Egypt, is that, like other markets, this stem cell is a promising segment, and there are several promising segments of stem cell market. We can focus on one or two or three of them, not everything. For example, the promising segment of stem cell market, which I suggest to Egypt, is that stem cell product or stem cell service, stem cell technology, and in the future we can have stem cell application. Stem cell product, like we can produce more human adult stem cell. We have a factory. We can be the factory of the world for human adult stem cell, embryonic stem cell, and other stem cell. We can be also stem cell banking. We can have more stem cell banking in Egypt, stem cell testing, isolation, and drug discovery. We can also have stem cell technology like stem cell acquisition, stem cell production, cryopreservation, and expansion and subculture. Hopefully in the future, this can be moved to the treatment of the patient in the term of stem cell application. So we can use that for the treatment of the patient. However, the bottom line, which I suggest to my mother country, is that stem cell research is a key. We can't jump to clinical trial without having basis of stem cell research on active stem cell research. So stem cell research is a key, and we need more development and investment and research, and then move from bench to clinic. It is bench to clinic, not the opposite. So finally, I'd like to acknowledge my research group, Carol, John, Karen, Emily, and my collaborator from University of Edinburgh, University of South California, and also from Caltech, Harvard, Boston, Columbia, and Paris, and our founder source. And thank you so much for your attention.
Thank you very much, Professor Ahmed, for an exciting, enlightening lecture. And as per the conference policies, we're going to allow the questions at the end of the session. If you can please stay with us till the end of the session, we'd truly appreciate this, because I know there are a lot of questions for you.
Yes, I'm happy to answer.
So allow me to present Dr. Minatullah El-Sirafi. She's an associate professor of genomics in Zuwail City for Science and Technology, and will be giving a lecture on transcriptional analysis of nuclear encoded mitochondrial genes in 8 neurodegenerative disorders, the analysis of 7 diseases in reference in Friedreich's ataxia.
Good afternoon, everyone. Okay, so today I would like to give a talk on a recent paper that we published. Okay. A recent paper that we published that describes the transcriptional analysis of nuclear encoded mitochondrial genes in 8 neurodegenerative disorders. And I actually chose this topic in particular because on this paper, if we can, just one second. Oh, no. Sorry. Oh, okay. Yes. Okay, I'll tell you later then why I chose this topic, but in general, our research interest includes yeast genetics and bioinformatics, and we also use human cell lines to confirm experiments that we do in yeast cells. So we work on different research areas, including investigation of DNA repair mechanisms, DNA damage bypass, and drug resistance mechanisms. And we also link our data to cancers and neurodegenerative disorders because usually the mechanisms that we study are linked to these diseases. In case of defects that happen in these mechanisms, usually the patients suffer from cancer or neurodegenerative disorders. Today I chose this paper in particular because this paper has 2 first authors actually, Mohammed Zayed and Reem Ali Al-Ghayj. And I chose it because Reem is a MUST graduate, and she graduated from the Faculty of Biotechnology in 2019. So I wanted also to give her the space to present 3 slides or 4. MUST أنا قلت أنا قلت إيه؟ أنا آسفة MUST I said that she's a MUST graduate. Okay, so she graduated from this university, so I wanted to give her the chance to present in front of you and to show you also the outcome of the students from the university. She's also currently doing her master's in my lab, and she's really one of the excellent students in the lab, to be honest. Okay. Yes, so today I'll talk about understanding diseases from the transcriptomic analysis point of view. So this is actually a picture that I really liked because it illustrates the process, although it talks about cancer, but it goes the same way for neurodegenerative disorders as well. So in the case of cancers, when we want to investigate the transcriptomic differences between patients and normal people, then we collect samples and we do RNA extraction. And then we perform RNA-seq analysis or microarray analysis in order to test which genes are transcriptionally dysregulated in these cells. And also through pathway analysis, we can identify which pathways are actually dysregulated in these patients. This is very important because when we want to personalize the therapy, it's important to understand which genes exactly are dysregulated in a specific patient in order to give this patient a proper drug, the right drug for him or her, and not just describe something that can be unspecific and also cause side effects more than being beneficial to the patient. So we applied the same approach to 8 neurodegenerative disorders. And in our analysis, so I just want to tell you that the neurodegenerative disorders are very complex because they can result from many different causes, genetic factors. Mitochondrial dysfunction is also linked to the neurodegeneration, ROS generation, accumulation of amyloid fibrils, for example, in Alzheimer's is a great cause. So there are many different factors. So we wanted to do a transcriptional analysis in order to see what kind of genes are not regulated well or are overexpressed or downregulated in comparison to normal people. And but with a focus on actually the mitochondrial dysfunction in particular, because we had wet lab data that supports that improving mitochondrial function can really help cells survive, and also using yeast as a model organism. So we focused on the mitochondrial dysfunction by studying the so-called nuclear encoded mitochondrial genes. So the mitochondria is very small, as we know, like 16,000 kb. So it codes only for 13 proteins. And this means that more than 1,500 genes are encoded by the nucleus, and then they get either imported only to the mitochondria, or actually most of the time to the nucleus, also maybe to the cytoplasm, depending on which signals do they have and where they function. So we have really a very long list of genes that act in the mitochondria, maybe besides other compartments also in the cell, but we wanted to investigate these genes in particular in these diseases. And that's because when mitochondrial dysfunction is linked to neurodegeneration, as we would see that alteration of mitochondrial dynamics happen, oxidative stress, the ATP biogenesis can be affected, and neuronal cell death happens also due to problems in the mitochondria. So, I'm sorry. So the aim of this work was to investigate the role of nuclear encoded mitochondrial genes in the pathogenesis of 8 neurodegenerative disorders in order to identify a new potential biomarkers. Let's say this is a bioinformatics study, so it points to some genes that can be later studied in the wet lab. And also, we wanted to see what kind of genes would appear for us in many different diseases. So what kind of genes are common between more than one disease? Because these can be like a major cause or underlying the development of the neurodegeneration. So these are the diseases that we tackled: Parkinson's, Alzheimer's, Creutzfeldt-Jakob disease, frontotemporal dementia, multiple sclerosis, Friedreich ataxia, amyotrophic lateral sclerosis, and Huntington disease. Okay, sorry. So the workflow, we first started with a Friedreich ataxia dataset, an RNA-seq dataset, because the Friedreich ataxia is one of the diseases that is very challenging, and it's not treated yet. There is no drug for this disease. So we started with it, and because also we found nice microarray datasets and RNA-seq datasets, so we thought we can use one for the beginning of the analysis and then complement it with the microarray data. In this case, we extracted first the nuclear encoded mitochondrial genes from the Friedreich ataxia dataset. And this was done through looking up in the literature and making a very proper list of nuclear encoded mitochondrial genes. We had almost like 1,600. And then we did an additional verification step by checking their localization on different databases and making sure that they actually have a high confidence that they go to the mitochondria. And then we performed the so-called WGCNA, which Shireen will explain. And based on this data, we did pathway enrichment analysis. She will also explain this part. And in parallel to this, we analyzed the transcriptional differences in the 8 diseases using, this time, a microarray dataset of Friedreich ataxia from adults and children instead of the RNA-seq, and also using microarray data for the multiple sclerosis and for Creutzfeldt-Jakob disease, and RNA-seq data for the rest of the diseases. We did the normalization steps and so on, and then we did differential gene expression analysis, and we tried to look what was common between this path and this path in order to focus on something that is really common. We're trying to narrow down as much as possible through checking the common between everything. These are the data sets that we used. They were all publicly available on the GEO database. And this is the number of patients and the control samples as well. Usually in the neurodegeneration, unlike cancer data sets, you don't find so many number of patients because you need to take the brain cells or the tissues after the patient die most of the time, unless it's a blood sample. And we always try to pick tissues instead of just blood samples. So it's difficult to have a huge number of samples for neurodegenerative disorders, actually, when you're working. So we were quite lucky to find, these are all the data set that we found that were appropriate with an okay number of samples, and that's why we chose them to proceed. Yes, and now Arim can explain the WGCNA. Oh, you have the, how did it?
WGCNA is a weighted gene correlation network expression analysis. Why we choose WGCNA? as for some reasons, no gene product can independently act in the cell. In addition to that, some tissues like brain are a complex hierarchical network and spatiotemporal, needed to link it to the structure and function. Moreover, some tests like the independent test generate hundred numbers of genes that are statistically significant, but statistically significant of the gene not usually related to the biological function. So using a network or building a network of modules of genes or clusters of the co-expressed genes help us to identify some ways. Firstly, we identify the function of some genes based on their co-expressed genes, which will be helpful in the case of unannotated or under-annotated genes. In addition to that, it helps us to identify multiple drug targets in the same pathway. In our data, the WGCNA clusters the NEM gene, which is a nuclear encoded mitochondrial genes, into 7 modules. We choose the 2 of these modules, which has a highly correlation coefficient. The highly correlation coefficient in the WGCNA represents that the list of genes are highly correlated to each other, which means that they are co-expressed or individual or involved in the same pathway. We then use the pathway enrichment analysis. The pathway enrichment analysis help us to identify the pathways that enriched in this list of genes. For example, the pathway enrichment analysis for the turquoise module identify some important pathways that are crucial for the mitochondrial dysfunction, like mitochondrial translation initiation, mitochondrial translation termination, and mitochondrial translation elongation. All of these pathways are well studied and linked to the dysfunction of the mitochondria and as well as the pathogenesis of the neurodegenerative disease. In addition to that, the pathway enrichment analysis for the brown module identify 3 pathways like NLRP3 inflammasome and peroxisomal protein import and the branched chain amino acid termination catabolism. All these pathways are well related to the pathogenesis of the Friedreich ataxia, but we want to know if this gene and these pathways are also involved in the pathogenesis of the other neurodegenerative diseases we studied. So we try to find the overlapping genes between the pathways and the modules and the differentially expressed genes of the other 8 diseases, including Friedreich ataxia, using 8 datasets.
So as Reem mentioned, we tried to narrow down as much as possible and take all the common genes by making like narrowed down list. So we focused on the genes that were in the brown and the turquoise modules, and then we further focused on those that were enriched in the pathways, and then we focused on the ones that were common between more than one disease of the 8 that we analyzed. And what was interesting is that we found, this is a summary of the important hits that we found that were not only common actually in one disease, but for example, you will find these 3 that we decided to focus on. So the Cas1 and Pickard were overexpressed in 4 different diseases, and only just in one it was downregulated. I'll explain later. And this was a very interesting hit. The DDIT4 was overexpressed in 6 different diseases. And the ribosomal proteins of the mitochondria, we found in general that they are dysregulated in the neurodegenerative disorders, and in 2 of the diseases, we found that it was downregulated. So we decided to focus then on this DDIT4 because it's very interesting that in all the patients, actually, 6 of the diseases, the patients had overexpression of this DDIT4. And the Creutzfeldt, the Alzheimer's, the Huntington, the Parkinson's, the frontotemporal dementia, and the multiple sclerosis. So that was very intriguing. So we read, like, of course, all what was in the literature to know if this is something that we found out or some information was already known. So we found that the DDIT4 was already reported to be very highly elevated, and it codes for a protein called RAD1 or RTP801. This name or that name are used, and they found it like really overexpressed in Parkinson's models before, and it was also associated with Alzheimer's and Huntington. I'm sorry. So in the Parkinson's and Huntington mice models, when you do downregulation of these genes, this help improves the phenotype of the mice models. In addition, the levels of the gene were also very high in the Alzheimer's model, and its knockdown improves the cognitive ability of the 7-month-old mice. So therefore, the literature concluded already that this can be a very promising target for neurodegenerative disorders. However, what was new in our paper is that we found also this overexpression in MS and Creutzfeldt-Jacobs disease and frontotemporal dementia. So this means that this is really a very important gene to study and to pay attention to in the future, because it's really common between many diseases. And then, oh no, sorry, now I have to put. مش عارفة هو اوكي انا حاسه ان هو بطاريته فدت او حاجه اوكي سوري كده جوه. Okay, so and then we focused then on this caspase 1 encoding gene and Pickard. Also, we found that they are overexpressed in most diseases except for this Friedreich ataxia adult data set. And that was also interesting because these two genes function in the so-called NLPR3 inflammasome pathway. So the caspase 1 is known to code for caspase, the casp1 gene codes for the casp1 protein, which is important for the activation of interleukin beta. So this causes inflammation. In general, it should be present in a low level inside the cells, but in the neuroinflammation, then this protein increases a lot in the cells and causing inflammation, which results then in cell death. Pickard also is involved in neuroinflammation. And previous data on Huntington brain samples, they detected this excessive activation of casp1, and inhibition of this protein slowed down the progression of the disease. So also there was an inhibitor that is FDA approved for other purposes. It was used in the case of the AD mice models, and it's called bilnacasan. And this inhibited also the caspase 1 activity and improved the phenotype of the mice models greatly. So even though the casp1 in our case was not actually overexpressed in Friedreich ataxia set as well, but previous articles reported its overexpression. And that is also important to point out, because this is a bioinformatic study based on a specific number of patient samples. So maybe if you have different patients, then you get slightly different results. So that's why we're trying to find genes that are really consistently behaving, or when we also focus on the personalized aspect of the analysis, we would find that we need to make sure that the patient we're trying to treat has this level of elevation in order to conclude that he can take a particular drug, something like this inhibitor, for example. So in general, we found also this MRPL1, like I said, any protein like involved in the mitochondrial protein translation and biogenesis, they were downregulated in many diseases. So here, this MRPL1, for example, was in Friedreich ataxia and MESPEC, it was downregulated. And this is also, this makes a lot of sense because the translation is very important in for the mitochondrial function, of course. And it's also known that it's important for the neural differentiation and the development, that these pathways happen like smoothly and without any disruption. So yeah, in general, so our data, because we combined 8 diseases, some of them were reported already in the literature, but by having like the analysis done in other diseases that were not reported before, we can suggest that these genes are really worth investigating further in the lab and on a personalized aspect. So the conclusion is that we obtained a narrowed down list of genes that could act in the future as biomarkers or drug targets. Yes, it's a bioinformatics study, so the data is very nice and promising for the wet lab people to check and to follow up on. However, you need to test it for different patients, different number of samples, different data sets, try it on different individuals, and also check each individual personally if this would work out for him or her to avoid the use of any wrong drug. So basically, we are pinpointing a set of genes that can help people investigate later, instead of looking at like big, massive lists that are produced through transcriptional analysis the normal way. By narrowing down this list through the WGCNA pathway enrichment analysis and checking also the common between multiple diseases. Yes, so at the end, I would like to thank Dr. Iman Badr, who's also shared the corresponding author on this article. She is a professor of biomedical sciences at Zoel City. And Reem, of course, and Mohammed Saadani, because they shared the first author on this article and worked very hard. Mohammed is also doing his PhD in the US now, and this was part of his graduation project. And Aya and Ala also, who worked very hard on this project, and Rana Mansour, I'm sorry, I forgot to write the name, but okay. And also the funding, of course, from Zoel City, L'Oreal, and ICGB, which contributed to making this article open access and readable by everyone. Thank you so much.
Thank you very much, Professor Minatullah. And let me just thank the mentor and the mentee. You are a great mentor for your students. Thank you very much. And again, questions are going to be in the end of the session. And we had a change of in the schedule. Now it's Professor Dr. Ahmed Zayed. He's a professor of medical physiology and biotechnology, College of Science, Cairo University, Egypt, and will be giving a talk on the B Venom and its active component, melatonin, synergistically potentiating the anti-cancer effect of sorafenib against hepatitis Hep G2 cells.
Good afternoon, everyone. It's my pleasure to be with you today to present one of the interesting titles under the name of B Venom and its active component melatonin synergistically potentiate the anti-cancer effect of sorafenib against Hep G2 cells. Perhaps first of all, I'd like to explain what's the concept of our research work, which actually depends on the fact that whenever you are dealing with cancer, you either start with radiotherapy or chemotherapy. Sometimes you extend to surgery. So there is a lot of interventions to treat cancer. And as you know, that chemotherapy is one of the harsh methods to treat cancer. In fact, my lab is concentrating in dealing with hepatocarcinoma or hepatocellular carcinoma, which is the main type of liver cancer, comprising almost 90% of the cases. And almost 800,000 dies every year from liver cancer or hepatocellular carcinoma. This is our publication released in 2021 in the Bioorganic Chemistry. Here's the content of my research proposal. This background and aim of work, experimental design, results, conclusion, graphical abstract, and recommendation. As you know, hepatocarcinoma comprises or being ranked as the sixth type of malignancy all over the world and the second cancer-associated death. But in Egypt, we consider that hepatocarcinoma is the first type of malignancy and the first type responsible for deaths. Actually, hepatocarcinoma is treated with one of the FDA-approved drugs, which is sorafenib, which is considered as a multi-kinase inhibitor. But as you know, toxic drugs or chemotherapeutic drugs has always many adverse side effects, including drug resistance and very, very harsh side effects on normal cells. Sorafenib actually acts by many mechanisms, but it inhibits, as I told you before, it's a multi-kinase inhibitor. It affects many pathways that actually controls many regulatory pathways, ending with apoptosis and preventing angiogenesis and so on. Sometimes it's also one of its mode of action is to stimulate oxidative stress. By introducing sorafenib, you usually interrupt the cell proliferation and angiogenesis. From the other side, as you know, there are recent approaches now looking for going back to the natural products, especially from herbal medicine and from animals, like venoms of some animals like bee, scorpions, spiders, and snakes. And we were very interesting to deal with B venom and its active component milletine. You know that B venom has been used for many decades to treat backbone pains and rheumatoid arthritis, many skin disorders. It has many applications as immunomodulation, treat neurodegenerative diseases, and anti-inflammatory, antiviral, and antibacterial. As concerning the chemical composition of B venom, the major component is peptides with small proteins, enzymes, amines, sugars, and minerals. And among the peptides, milletine as the active component comprises almost 30 to 60% with other constituents. Here is the major composition of milletine, which is about 26 amino acids. The aim of my work is to study the potential use of either the crude venom, B venom, or its active milletine, either singly or in combination with sorafenib to treat cancer, in a trial to avoid the very high rush side effects of sorafenib. So we'll study the single and combined effects of milletine, either with the crude venom or with milletine, and check for some molecular studies, and starting with molecular docking as in silico study. Okay. Of course, we will check its effects on both normal cells and hepatocellular carcinoma cells, which is called Hib2 cells, and to make a combination of both, I mean the sorafenib and the milletine or the crude venom, and to check if this combination could help to reduce the harsh side effects of sorafenib. Here the workflow of our work to check for the molecular docking. We have choose some protein receptors, serial receptors, and see what's the interaction of milletine with these serial receptors. I mean, whenever you get a high affinity, this means that the milletine has its binding affinity, a high binding affinity with this type of receptors. We made cell culture for the Hib2 cells and the normal human liver cells, SLE2. Okay, and then we analyze this cytotoxic effect by MTT assay. And after that, we got the estimation of the IC50s. Okay, and we choose to do some combination with 24 non-constant combinations, depending on the data from the first experiments with single cytotoxic effect. And we usually used the CompuSen software for estimation of the combination index. Whenever you get a combination index of less than 1, this means that there is a synergistic effect, and if it is more than 1, so there is an antagonistic effect. Okay, and we choose only 2 concentrations from these combinations to make the downstream analysis for some very interesting selected genes, usually concerned with apoptosis, angiogenesis, metastasis, cell cycle regulation, and so on. So my result will be divided into 2 parts, I mean the in silico part and the in vitro part. For the in silico, here are the interaction of the milletine with different cellular proteins, and we got the results that there is a high affinity with almost of these receptor proteins, except for the vascular endothelial growth factor receptor. Whenever we get a negative value for the combination energy or binding energy, this means it is high affinity. And whenever you get a positive binding energy, this means that there is a less affinity for this type of receptors. So we got very good results with almost all the cellular proteins except for vascular endothelial growth factor receptor. Here the interaction with many protein receptors. And now for the in vitro study with the effect or cytotoxic effect of each individual component. Here, as you see here, that's sorafenib. Sorafenib has an IC50 of 3.9 microgram per milliliter in comparison with the normal cells. Just a minute. Here the effect on the normal cells 3.95 versus the effect of B Venom, which is 95.73 microgram per milliliter for B Venom, the crude venom, and Militin or 78.38, which means by some way that the B Venom and Militin are very safe on normal cells in comparison with sorafenib. As you might know, that sorafenib has very harsh side effects and affect normal cells as well as the cancer cells. As concerns hepatocellular carcinoma, you see that's IC50 for for sorafenib was 4.15, but for B Venom is 2.8, and for Militin is 1.4, which means that either B Venom, the crude venom, or Militin is considerably highly toxic against cancer cells, but not on normal cells. Back to this slide of the normal, the IC50 is 95.7 and 78.3. Here are the combinations of Militin and B Venom, crude B Venom with sorafenib in, as I said before, 24 non-constant combinations. And in the yellow color here, there's a combination with 0.75. This is 3 to 4 of the value of the IC50, combined with 1.5 for B Venom, okay? And you got the cytotoxic effect is around 95%, and almost cells were died. And the combination index here is 0.01, which means that there is a synergistic effect. On the other hand, this is a combination of Militin with sorafenib, and we chose this combination, which contains also the least amount of sorafenib, 0.75. This 3/4 of the value of the IC50 was 3 times the IC50 with Militin, and we also get the same CI50 combination index, which is less than 1, which means that there is a synergistic effect. Okay, so we choose to do some downstream analysis for some genes, P53, BUX, caspase, and caspase 9, PTN as apoptotic markers, and we find here there is an increase in the expression after using these combination, as I selected before, the yellow and blue one. Okay, there is a high significant increase versus the normal and single effect for all these apoptotic markers. But as concerns BCL2, which is anti-apoptotic, we got a down regulation, okay, which by some way means that there is an intention to start apoptosis. Here are some genes related with cell cycle regulation, metastasis, hypoxia, inflammation, necrosis factor kappa beta, and metastasis or angiogenesis, the vascular endothelial growth factor. Here we got very interesting results that we got a down regulation of all these genes, okay, which means that the combination of sorafenib with either Militin or the crude B Venom has a potential effect to inhibit the genes. Then we switch to the picture or the what the effects of single and combined effects of melitine, sorafenib, with sorafenib, I mean, or crude venom with sorafenib on the stress markers. Of course, MDA has been increased significantly in the 3 types of the sorafenib individually or in combination with both venom and melitine. But on the other hand, SOD, catalase, and Defects, there was some changes, but not so much effect, except for sorafenib, which may implies us some way that combination of sorafenib with melitine or crude venom increase the oxidative stress for the cells. Here are also some type of flow cytometric analysis for the cell cycle analysis, and we got the conclusion that either single effect or combined effect always make cell cycle arrest at the G2M phase, and which again reflects the potential of either single or combined effect to arrest the cells during the G2 phase. So from both results of in silico and in vitro studies, we came to the conclusion that we can use sorafenib in combination with melitine or crude venom to reduce the very harsh side effects of sorafenib. As I pointed before, that the sorafenib has a very high cytotoxic effect on both normal cells and cancer cells. Of course, whenever you are dealing with the crude venom and the active component or isolated and purified component, you choose to deal with the purified components because it's a single compound, and as we know that it's a peptide, so we can make a mass production of this protein, okay, to be combined with sorafenib to achieve apoptosis and inhibit cell proliferation, angiogenesis, metastasis, and so on. Here are a graphical abstracts and the summary of all of our results. The blue one means that's down upregulation, and the red one is for down regulation. And as we see here, we have interrupted apoptosis. We increased the rate of apoptosis, okay, and we arrested cell cycle and inhibit metastasis, immunomodulation, oxygen supply, and angiogenesis. As a recommendation, the present data may open a new avenue in recent approaches of hepatocellular carcinoma treatment using natural products, either by B venom, the crude venom, or B melitine as adjuvant, or being used alone as alternative therapy. And B venom and melitine almost have the same effect, but we recommend melitine to be used in the treatment of hepatocellular carcinoma as a pure and effective form. Thank you.
Thank you very much, Professor Zayed. Now, this is the last presentation of the Professor Dr. Mohamed Egazi, Professor of Cardiology, Cairo University, Egypt. And the presentation is, the title of the presentation is Cardiac Glue, Is it a Dream or True? Please, Professor.
بتشتغل ازاي كده كده هنا هنا كده
ده كده يمين ده شمال لو حضرتك محتاج بوينتر عشان بس ليزر لان هو عيني هتسقط بس انت بس ليزر لحضرتك
طيب
وانا جنب حضرتك هعمل حاجه
ماشي السلام عليكم وكل سنة وانتم طيبين بمناسبة المؤتمر الجميل ده. Good afternoon, and I appreciate my attendance here in front of all these eminent scientists and the chairpersons. It's my pleasure to introduce my very simple talk about the cardiac glue after all this heavy, yes, it is heavy scientific words from the previous colleague. I hope that I will be like in Egypt يقول أكون ضيف خفيف عليكم. دكتور علا, it's my pleasure. My idea about the novel regenerative solution to all the cardiac defects. What is the congenital cardiac defects? That's introduction. Congenital cardiac defects are abnormalities of the heart structure and function caused by abnormal disorder heart development before birth. The prevalence of congenital heart disease in Egypt is 1.1% of live birth population. This is my research done in 2017. I did it from a lot of governorate, like from Aswan and Alexandria and Luhat and Mansoura, Daqahliya and Sharqiya. I found that it is 1.1%. That's why I'm so interested about the congenital heart disease, because if we don't treat it well, it will cause a complication. The most commonly, the ASD is ostium secundum defect, occur in 5 to 10% of all congenital heart. About 50% of children with congenital heart have ASD as a part of cardiac defects. Isolated BDA, patent ductus arteriosus, account 10% of all congenital heart disease. And ventricular septal defect is currently the most common, is 20 of all such defects. As populations ages, VSDs have become the second most common congenital heart disease. What is the complication? If you don't treat it well, long-term complication can include congestive heart failure, infective endocarditis, arrhythmia, pulmonary hypertension, and Eisenmenger syndrome. The old management of the congenital heart is the medication. The medication in the previous century, medical therapy was mainly based on the pathophysiological consolidation or parallel with acquired heart disease, like anti-failure measure. All of you is familiar with anti-failure measure and anti-subacute bacterial endocarditis and anti-arrhythmia. Over the past decades, a large amount of research has been performed to detect the benefit of pharmaceutical intervention in patients with congenital heart disease. Specific population have been studied more than anti-pulmonary hypertension like VAN. Now, the second tool of the treatment of the congenital heart is the surgery. Since ancient decades, surgery has been the cornerstone in the treatment of patients with congenital heart. Since the first successful ligation of the BDA in 1938, large-scale intracardiac repair started after the development of cardiopulmonary bypass technology in 1950s. This is the operation. The first successful surgical of the ASD was performed in 1949. This is the ASD. The possibility of deep hypothermia with a circulatory arrest in 1970 has made cardiac surgery as a terminal success in the reduction of morbidity and morbidity patients born in congenital heart. The cardiac intervention, this is the second or the third old management, and now become old because now it is we started the catheter CAS in, I think, in Egypt in 1992 by Dr. Rosemary and the team of Dr. Magdi Aoub. He is I'm the honor to be a student of Dr. Magdi Aoub, because this man he did a lot, and he will doing a lot for the cardiac patient and doctors in Egypt and all over the world. With the development of the catheter techniques, percutaneous intervention have replaced some forms of surgery, but also extended therapeutic arsenal. The first transcatheter closure of the patent ductus arteriosus was done in 1967. Now this is a BDA devices. This is what we use in the cath lab. Now this is the operation of the BDA. This my case. Now if you will see the cath, we inject the aorta and then we introduce the cath by the here by the device to close the BDA. Now we close it well. And here, this is the apparatus after deployment successfully. After that, we started in 1975 by Rashkin, the ASD closure. This is the ASD devices. We have a lot now of ASD devices closure. Now, this is my patient also. Now, we employed this apparatus or the device is Amplatzer device to close the ASD. Now we're successful here. We remove the cath and deployment well, and the patient become normal after 2 or 3 hours. Now, percutaneous pediatric catheter intervention attempts to close defects extend to all the type of ventricular septal defects. The very first percutaneous VSD is 2013. This is a type of a lot of type of VSDs. Now, a large collection of devices has been developed and tested to closure rate. Most popular devices consist of two disc system. This is a VSD devices according to the position, and this is another group according to the position. Now, this is my patient. He has VSD, as you see here. We inject in the left ventricle, and this is a leakage, and we use it. This is the Amplatzer also. Now we close it completely without any residual. Again, this is another case here also. Okay. Now, transcatheter embolization has become the preferred treatment for aortopulmonary collateral also, and systemic and venous fistula and venous collaterals, and also coronary artery fistula, replacing the surgical intervention. Now, this is a lot of types of the AV fistula. Again, here the coronary fistula. If you see here, we closed it carefully. And this is MAPCA. Also, this is the collaterals. Now, this is my patient. I use this plug to close the collaterals here. Now, additional developments have been deployed in hybrid procedure. Hybrid is a combination between surgery and the cath. Now, what is the cardiac glue? Is it dream or true? When I was in Morocco, this is Casablanca Mosque, very famous Al-Hassan al-Thani Mosque. I found there is a salamander. I did the pilot study, what you see is fikra magnuna. I found the salamander in Morocco, and this salamander, if we cut anything, any part of it, it will be regenerate again, even the piece of heart. Now I do the pilot study, very simple there. I did 2 or 3 sacrifice of the salamander, and I take piece from the pericardium of the salamander. I do the homogenate in the BPS, phosphate buffer saline, and I centrifuge it by the ultra micro 0.22 ultra micro, and then I take this extract and I inject in vitro and in vivo. But by close observation, I found that the rat I injected is died or arrested after 3 days, and the in vitro, the piece of the heart is sloughed and become necrotic. Now I return back to Egypt. This is my colleague, Dr. Hassan Sobhi, and this is Dr. Said Fahim. And this is Dr. Ahmed, all of us familiar with him, and Dr. Mamdouh. And this is our juniors. I appreciate all of them. Now the team, our team is Dr. Safa Nasr, Ahmed Zayed, Hassan Sobhi, Said Fahim, Mamdouh Nassar, Sameh, Magd Abu Sofia, these from Morocco, and Moorman, this from Holland, and lastly, my name, Higazi. Now, what is salamander? This one piece of salamander. This is a lot of species of salamander. Salamander, it is one of the vertebrate urodele amphibian tetrapods. It is the only one has the ability to regenerate complex structures such as limbs, spinal cord, and the cardiac tissue throughout their lives. The majority of salamanders of Africa are endemic in northwestern Africa, in Morocco, in Tunisia, in Algeria, the north coast of Tunisia, also in the coastal mountain in North Algeria. In Egypt, it was some little in the northwest coast in few colonies. Its natural habitat are temperate forest, river, caves. The family of species Salamanderi is the one live in the northwest Africa. Salamander fibroblast proliferate, giving rise to the multipotent mesenchymal growth zone plastima cells, which is responsible for the tissue regeneration process. Its regeneration rule and mechanism is still unclear. Till now, it is unclear. Now, the injury of the salamander cardiomyocyte leading to enhance the release of species transgenic intermediate factor. The intermediate factor polarize macrophage, and then the micropeptides containing these factors could promote the endogenous cardiac regeneration using multiple emulsion solvent evaporatant technique. The growth factors which require to promote cardiac tissue regeneration have induced angiogenesis process, which supply the oxygen, the micronutrient to the cardiac tissue, maintains the biological function. What is the clinical impact? This glue will use what we call the cardiac glue. We're used to management of the congenital heart defect like ASD, VSD, and BDA instead of post-surgical and CAS. Plan of our project. The first is detection, the second is reduction, the third isolation of the salamander growth and regeneration factors from its serum and the cardiac tissue using the following different techniques: cellular and molecular biology, stem cell biology, tissue engineering technique, immune histochemical investigation, histological studies, microscopic investigation, and the histobiological study, electron microscopic study, scanning and transmission techniques to detect the ultrastructural changes in the salamander and the experimental animal. Biochemical studies of the salamander growth and regenerative factors using chromatographic techniques and PCR real-time Western blot analysis, protein fraction, and proteometric analysis. Surgical biopsy of the salamander cardiac whole pericardium via open heart surgery under general anesthesia procedure, then biopsy will operated upon by previously mentioned technique. These factors which present in salamander plastima we found in the literature. It is protein, BROD1, retinic acid, GP1, FGF1, and MIGF1, and HMGB1, and neuroglin 1, and CD59. Prepare this factor to be in the injectable form to inject them into the experimental animal cardiac tissue. We started by the rat, and then after that we'll use the guinea pigs and dogs, and then monkeys sequentially. The concept. The aim of our research is to discover new matrix like glue, which depend upon the salamander growth factor and its regenerative mechanism. The science of nanotechnology has introduced different biocompatible natural material like ketazone and collagen that can be fabricated in the form of nanomembrane, and these membranes can be loaded with many drugs like transcription and the growth factor together with mesenchymal cells and serve as a matrix for tissue engineering. This is the role of Dr. Ahmed. The science of bioinformatics can also help to identify human proteins which, like the growth factors, as Salamander, depending on the gene and the protein sequence. We hypothesize that a type of cardiac matrix like glue could be made via the technology of tissue engineering and nanotechnology. If the generation has success, after implantation, the growth factor and regenerative stem cells in the experimental animals' cardiac tissue, we will use human cardiac tissue under the ethical international research rules and laws. Requirements: we need experimental animals, cage and feeding care. We need kits and extraction procedure laboratory. We need electron microscopic team. We need immunohistochemistry lab, and all of them we are now belong to our team. Molecular biology, tissue culture and tissue engineering, and genome study in genetic laboratory, stem cells techniques, surgical team and anesthesia team. Participating faculties: Faculty of Medicine, Faculty of Science, Cairo University, Faculty of African Research and Studies, NRC Cairo, Faculty of Medicine, Casablanca, Morocco, NDCRT, and the Holland. Take-home message: congenital cardiac defects are ancient old disease. Till now, there is no unique specific solitary solution. For long decades, the surgery had considered the cornerstone of the management. The novel researches are directed and deviated the management, the way of the management toward the interventional cardiac catheterization, the hyper technique taking the role in congenital heart management. And lastly, I hope that my dream about the cardiac glue to be applied in congenital heart disease management and soon become true. Thank you very much.
Thank you very much, Professor Mohammed, for a truly, changing the practice, idea. I hope it really does make a difference in our patients. Thank you very much.
Thank you.
So the floor is open for discussion. We have 5 minutes for questions.
Sorry. My question is for Dr. Khorshid.
Yes.
Which I understood from your excellent presentation that your work was stressed on the limited to the epithelial growth factor receptor mutations, right?
No, well, for lung cancer, we have 8 mutations that we currently give target treatments to. I just give an example on EGFR mutation because
But did you do the microarrays test? You know that, as you know, thousands of genes controlling the carcinogenesis related to different groups of genes, such as oncogenes, tumor pathogens, and others. So we consider every patient, cancer patient, is an individual cancer, which is different from the others. At the same, if you have 5 or 10 breast cancer patients, everyone has different mutations from the other group, right?
Yes, that's very true.
So when you have to have a specific mutation to be considered as a personalized medicine, you have to do a genomic analysis first.
So
you can't use common treatment or common, you can not detect a common mutation in different cancer patients.
and
Am I
that's
right?
where NGS is going to be very important. Now, for frontline treatment, we have standards of care. EGFR mutation, we have different exon mutations. And we have different targets for the exon mutations. And we have the ALK mutations, and we have the RET, and we have the ROS, and we have, so 8 different mutations. We needed to standardize that. You're absolutely right that for every patient, we're going to be having personalized treatment for them. However, now we need to have standard treatments for those patients. 63% of our lung cancer patients do have oncogenic-driven mutation. Now, we started knowing this when we started to look at the molecular analysis. And we started with only 2 target treatments for the ALK and for the EGFR mutations. And then we added 8 more. So year by year, we discover more drugs. Now, it's interesting that we discover the mutation, but it's more important to have drugs that will target these mutations. So for now, what we have is we have 8 mutations, we have target treatments for those mutations, and this changed the face of treatment of lung cancer.
But you know that carcinogenesis is a multi-stages process.
Yes.
So you have to have a molecular diagnosis for more than one mutation in the same patient.
This is why, in the beginning, the mutation is going to be exclusive. So if you have an EGFR mutation, you're not going to have an ALK mutation, for example. However, at resistance, this is when, or at progression, this is where you need to retest your patient again, because he's most likely going to develop a new mutation, as I showed in my presentation. So yes, you're absolutely right. But this is the standard that we do now. We test for the 8 mutations, and when you do have the mutation, you need to monitor that patient, and you need to look for the resistance, because he will develop new mutations. That is very true.
Yes.
So you're absolutely right. Thank you.
Yeah, I have a small question, second question. What is the difference between the liquid biopsy and the pleural effusion in the case of lung cancer?
So again, this is all liquid biopsies. You can have the liquid biopsies from the blood, you can have it from the CSF, you can have it from urine, you can have it from the effusion too. However, the most common that we use is the blood, because it's very easy to get, and it is you will have more circulating tumor cells and circulating tumor DNA in the blood. Yet if you have a patient with, for example, CNS metastasis,
I understand
it's very, let me just continue. So it's the detection of circulating free DNA is going to be very minimal if you have CNS metastasis. This is where you have to go for CSF cytology. If you have that patient who has stage 4 pleural effusion, for example, then maybe it's easier. If you cannot take a biopsy, then it's going to be much easier to take cytology from the pleural effusion and try to find the circulating tumor DNA. However, not all of your patients are going to be having pleural effusion. So this is also one of the liquid biopsies that we can use, but only for those patients who have pleural effusion. Yeah.
Thank you so much.
You're welcome.
I have one question, please.
Yes, for me too.
For you. I have been working on cell-free DNA. I did examination on tongue carcinoma with one of my students in Jizahri on how to detect the tongue carcinoma or squamous cell carcinoma from blood. But the problem is searching for such type of cells in the whole blood is a very tedious work. So what do you advise?
Well, tongue cancer is, I don't think it's that of a shedder for circulating free DNA, is it? This is why I showed in my presentation that not all cancer cells are going to be shedding the circulating free DNA.
Depending on the stage.
So it depends on
stage.
And the tumor, your tumor, it depends on the stage, it depends on the bulk of the disease. and your assessment methods. So we were looking with NGS. Is that clear?
No,
Is that
methylation pattern.
okay? And how sensitive would that be compared to?
That was the problem.
so it's very important to really choose the method that you're going to be detecting the circulating free DNA with, because false negative results are the biggest problem that we see with the liquid biopsies.
So the NGS is the only solution.
NGS is the best, yes, because you'll be able to look at, as was said before, you'll be able to pick up all the mutations that could be present.
Sure.
And especially if we're looking at resistance, then this is what we need to do.
Okay, very nice. Can I have one single question for Dr. Higazi, please? Yes. Professor Higazi, you mentioned that you use you are going to work on Salamander just to regenerate the cardiac muscle cells. My advice as a geneticist, I would like to go forward and go deeper to search the genes that is responsible for regenerating this tissue. Do you have any plan to search for genes that are related to that?
Yeah, this is a very good question. Me and my colleagues, Dr. Ahmed and Dr. Mamdouh and Dr. Magid, and also Dr. Norman from Holland, all of us agree with you. That's why we have to know the genome, yes, and we have to know the gene, and we have to know the difference between the salamander gene and the human gene, because we plan to use this glue to treat the human, not the animals. That's why we have to do a deep, deep study and more deep investigation about this very important issue.
So we have to delegate one of our guys to join your research team.
Yeah, of course, it's my pleasure.
Thank you, Professor Khorshid. Thank you.
Thank you. Thank you very much. So, any more questions? So, if there are no more questions, I would just like to again thank my co-chairman, Professor Soli. Thank you very much. And I would like again to thank Professor Hussein for his very kind invitation. It was quite an honor to be here. Thank you very much. Thank you. I enjoyed it very much. Thank you. So now is the lunch break, and there is also going to be a poster session.